It's not that what we discovered provides a cure for cancer, but what it does is it provides an intellectual framework which helps you think about the ways you can treat and cure cancer.
This is the James Cancer-Free World podcast. I'm Steve Wartenberg, and today is a special episode with two guests, including our first ever Nobel Prize winner, and that's Sir Paul Nurse, a geneticist who first identified a key gene that controls how cells grow. And this was a huge step forward in understanding how cancer cells grow and led to the development of some new and better treatments. Sir Paul has served as chair of the Department of Microbiology at the University of Oxford, Director General of the Imperial Cancer Research Fund, President of Rockefeller University in New York City, Director and Chief Executive of the Francis Crick Institute in London, and President of the Royal Society. He was awarded the Nobel Prize in Physiology or Medicine in 2001, along with Leland Hartwell and Tim Hunt. Sir Paul is here in Columbus as the recipient of the 26th Annual Herbert and Maxine Block Memorial Lectureship Award for Distinguished Achievement in Cancer. And in addition to delivering a lecture, the Block Lectureship also includes a collaboration between Sir Paul and someone here at the James, and that mentee is Maxine, an up-and-coming young scientist and an assistant professor in the Department of Biological Chemistry and Pharmacology. Maxine will join us in the second half of this episode, but first we'll spend some time with Paul. Welcome to Columbus and welcome to the James Cancer-Free World podcast.
It's a real pleasure to be here, Steve. It's not my first time, but I haven't visited for several years, and you've put on beautiful weather for my visit.
We did it just for you, so don't come back next week. It's going to be 102 degrees. I've been doing a little research and listening to some of your podcasts and reading some things, and two things you said, more philosophical than science, I was really struck by because I think they're great advice for anyone, scientists, podcasters, anyone. And let me tell you the two things, and you can explain them and how that's guided your research in life. And that the first one is you must not work too hard. And the second one is failure is not a disaster. So tell me about how you developed these keys to life.
Why do I say you shouldn't work too hard? I'll tell you the reason is because one of the major blocks in scientific discovery, which is what we're trying to do, discover new things, is that the mind begins to run on railway tracks. You
Yes.
get a certain view of how things work. You design then experiments to actually try and confirm that, and you can't escape it. And one way to escape it is, frankly, to stop thinking about it. Go away on holiday, go for a walk. We were talking about going up hills and mountains. Go up a hill and mountain, forget about your research. Then you come back fresh and you look at things a bit differently, and you can jump the railway track and realize there's different ways of explaining that. And the second thing about, failure is not necessarily failure. It's because one thing that people don't realize is that a very important way of achieving scientific progress is actually to show that something is wrong. It's actually quite difficult to show things are right, maybe impossible. So when you have your favorite idea, your favorite hypothesis, you should attack it. And when you show that it's wrong, you've actually made an advance. You've cleared away the scrub, if you like, and you can think of new ways of finding out how it really works.
Do you find that second one is failure is not a disaster, that some people are so stuck on an idea they refuse to give up even when the evidence is that it's wrong, and then that can slow them down?
That slows them down, but also there's something else. They get depressed.
Oh, yeah.
They get depressed because you do experiments, it doesn't confirm what you think. And they think, we've made no progress. The reality is you have made progress because you've cleared around ideas that are actually not the right ideas. And that helps you proceed to finding ones which are better.
So there you go. When you find out you've made a failure, go for a hike, get it out of your mind, come back fresh, and start on the next step.
You've got the first stages there of being a good scientist.
Whew, I don't know about that. And let's get into science. You're known for being one of the pioneers of discovering how cells grow. So fill us in on what you actually discovered, and then we'll get into what I think is important is understanding cell division, particularly cell division that gets out of control is the basis of cancer research. So fill us in on what you discovered.
First thing to know is that every living thing is either a single cell or a collection of cells. You are, I am. And therefore, all growth and development of every living thing is based on the cell. And what cells do is they grow, but then they divide from 1 to 2 and 2 to 4, 4 to 8. And it's the second of those processes, which is fundamental to growth, which I've researched mainly, in fact, most of my life. And this is a fundamental process. You're starting with a single cell, and that single cell divides into 2. Now, for that to happen in a successful way, you have to ensure that all the components in that cell that are essential for its survival get copied and distributed into the 2 newly divided cells. Otherwise, they lose something and they're not going to survive. And the critical component is DNA, the hereditary material. And there's 2 processes that are critical to every cell division process. One is the copying of that DNA, the double helix and unraveling and so on, and that occurs at the beginning of the cycle or somewhere near the beginning. It's called S phase for synthesis. And then at the end of the cycle, mitosis and cell division, where the replicated DNA is segregated through the chromosomes that separate at that time into the 2 newly divided cells.
So it's in the middle, and as they separate, that separates too.
Exactly. They've been copied earlier on, and then the 2 copies have got to be separated precisely. And that has to be controlled, and really my research has been focused on what brings about those events and what brings about, if you like, the temporal order, the order in time that needs to take place to get a successful cell division. And it's important to realise that process, I've said it already, is not only fundamental to the growth of every living thing, as you said in your introduction, but also when it goes wrong, when you get cell division that goes out of control, then that is the basis of cancer. Sometimes we want our cells to divide. You cut yourself, then you want the cells around that wound to divide and fill up the wound. But if you have cells out of control because they're cancerous, you can get a tumour, blockage of a function, an organ not working properly, and this is the basis of all cancers. So understanding this process is important for understanding growth and development, but it's important also for cancer. Actually, there's a second reason for cancer too, because cancer is caused when you have damaged DNA, genes get damaged. And so the process, as I said, the copying of DNA, the segregating of the DNA during mitosis and cell division, if they're not done precisely in the right order and the right way, that can damage genes and is a cause of cancer.
So they're reproducing wrong.
Exactly right.
So, and take us back, this is, I'm guessing, the 80s, when you started on this path. You weren't thinking cancer, were you? You were just thinking cell division and not necessarily this is what we need to understand to cure cancer.
Well, I'm even older than I perhaps look. It was the 70s when it actually began. But you're right that the 80s were particularly important towards the end of the 80s. I was interested, I thought a lot when I finished my PhD about what I should study. Because being a scientist is actually a lot of work. Yeah. There's a lot of failure, as I've already explained. Yeah. And if you're going to do this, which I'm totally dedicated and driven by it, then at least you should be studying something important. And I tried to think, what is an important basic process? And I've just explained, what is more fundamental than the process that is the basis of the growth and development of all living things? And not only that, relevant for cancer as well. So I decided to work on it, but not to try and, if you like, cure cancer, but to just understand, how can I put it, the principles behind dividing cells and what ensures the genomes are properly separated and so on, which are critical for bringing about cancer, because I thought that by understanding it, we would inform a basic biological process, and it has implications for cancer and other diseases.
Yeah, before you can cure something, you need to understand it on a biological and genetic basis, and that's what you did.
And sometimes that's forgotten. Sometimes because everybody is rushing so quickly to look for cures and treatments, which we have to do.
Yeah.
They sometimes think they know enough already. And sometimes I hear that people say, oh, we know how all this works. It is almost always never true. They're arrogant when they say that. What we have to do is to realize the whole scientific process is one of discovery, constant discovery of new things, and then to bring societal goods, by which I mean clinical advances or commercial ones, and then seeing which of those ideas, that knowledge, can be turned to the public good. But you can't just focus on one or the other, because the whole thing just falls apart.
Yeah, that's a great point that I have thought of, that basic science, fundamental science, is the foundation of everything. And even as you learn this much, there's still this much more to learn. And like you said, you can never be satisfied and think you know everything, because that's when you get at it, that's when you make mistakes.
You used a nice word there, foundation. There's a metaphor which might be useful here, that if you build a building taller and taller, and you're not based on firm foundations, it's only a matter of time before it falls over. So if you think the building of the height of the building is the application of the science, without the firm foundations and growing firm foundations, you cannot push that building higher.
And then even when you do that, you still need to renovate and update with new science as it comes along.
As you want to go higher, the foundations have to be better and bigger.
Wow. So help me understand how thousands of scientists all over the world took your fundamental research of cell growth and division, good and bad, what goes right, what goes wrong, and how that has helped lead to the modern cancer science of immunotherapy and CAR T-cell therapy and things like that.
Well, the first thing to realize is that actually I did this work in yeast. Now, who cares about yeast? I'm sure you don't care about yeast very much. You might,
unless I'm making bread.
Yeah, making bread or making beer, making wine. But it isn't your high up in your interests. But the beauty of working on something like yeast is it is simple. And when you're dealing with a complex process, which leads the division of one cell to two, there's a lot of complexities. And by working on a simplified version of it, you can actually work out the principles behind those processes. So then when you move to more complicated organisms, like cells in our body, you have a foundation to work on. So I and my colleagues and other laboratories worked on simple organisms, 2 different sorts of yeast, sea urchins, to work out what was happening.
What made you think of yeast?
Because yeast is amazingly good at as an experimental organism.
So this is something other scientists have been using.
Yes, they've been using it for quite a while before certainly I started. It's got they have 5,000 genes as against 25,000 genes. Much less complicated. All they do is grow and divide, which is actually what I was interested in. They don't make ears, and they don't make noses, they don't make eyes, all of which are complex developmental things on top of the basic process, and can be confusing. Because if you work on a cell, which in a human body is making all sorts of different tissues and cell types, that is distracting from actually the thing you're trying to study. But why I emphasize that is it was important to show that the genes that controlled this process, it's called the cell cycle, by the way, were the same in humans. And that my lab also did in an experiment which had no right to work, if I can be completely blunt with you. We had a yeast cell which couldn't divide because it was defective in the critical gene. We called it CDC2, but let's not worry too much about the names, okay? So it was defective. And we took the first human gene library that was made that could be used for this sort of purpose, and we used it in a way that nobody else had thought of. Basically, metaphorically, I sprinkled the genes on the defective yeast cells, arguing that if there was an equivalent gene in human DNA, and if a yeast cell that couldn't divide because it was defective in that particular gene took it up, and if that human gene could work in the yeast cell, then the yeast cell would now be able to grow and divide, produce a colony on an agar plate, and we could then isolate back the human gene. That experiment had no right to work. If you think about it, yeast and humans probably shared the last common ancestor probably 1,500 million years ago. I'll just say that again, 1,500 million years ago. Dinosaurs went extinct at 65 million years ago, just a tiny tick in time in comparison.
So there was just some water sprinkled around the planet. That's when they combined.
It's amazing. If you think about it, what it means is that once this system was set up, it remained the same for all that length of time. But for that experiment to work, it did demand that was the case, which is why most people, bluntly, including myself and my colleague called Melanie Lee, who actually did it with me, were thought to be a bit crazy about doing it. And I thought we'll give it a month or two and then probably move on to something else. But actually, it worked.
So does this prove that humans and yeast come from the same place all those millions of years ago, that we were once related?
Absolutely.
Yeah. So
you're related to a yeast, and that's the reason why the work is relevant to you. That's absolutely right.
And all human beings are related.
And all human beings.
We're all the same.
For that matter, because yeast is one of the simplest organisms on the planet, humans, we like to think, one of the most complicated ones, everything in between. So everything you look out of the window at the tree, at insects, at a sheep, all have cells controlled in the same way.
So once you showed this, what did that mean? What did that lead to?
So, two things, I've already briefly mentioned them. One is cancer's caused by cells growing out of control. So ultimately, cancer defects will have to influence that mechanism for cells to divide out of control. The way I like to think about it is that what we discovered is like the core mechanism for controlling cell division. Then there's a periphery around it, which is all the cancer controls, which might be going wrong for all sorts of reasons to do with the complexities of producing you sitting here, Steve. But they all have to end up activating this program, otherwise you will not get cancer. So it's not that what we discovered provides a cure for cancer, but what it does is it provides an intellectual framework which helps you think about the ways you can treat and cure cancer. It's an essential for the, if you like, the intellectual fabric for what you're doing. That's one thing.
It's the roadmap for curing cancer.
Without it, you don't know where you're going.
Yeah,
it's a sort of roadmap. And the second thing is, which is a bit closer, is to do with what's sometimes called genomic instability. That is where there's damage to genes, they either get mutated or rearranged or not separated properly, which are all things that have to occur successfully in a normal cell cycle. So by understanding those processes, you can also understand something about how cancer is formed. So it's providing, I repeat, the intellectual framework to think about the problem.
Wow. So that was 30, 40 years ago. You showed this.
Well, it started 30, 40 years ago. I mean, you can tell that from the colour of my hair, of course. But it's gradually built up over the decades.
That's what I was going to ask. How has this progressed, and where are you now?
Well, how it's progressed is that basic model's been shown to be correct. But some of the details, some of the more important details, are probably not correct. And so my lab spends much of its time in testing some of the assumptions that were made, including from my lab 25 years ago, and challenging them. Back to what we were talking about at the beginning. Always challenge your own ideas. In fact, I put it more sort of poetically. You should be your ideas, you should be their greatest enemy. Their greatest enemy. And we are now changing, and in fact I'm giving a talk here about some of the changes I think we have to now adapt to understanding how this works. Not undermining the fundamental principles, but new principles that we should take care of.
So it's refining and learning more.
Finding, learning more, and keeping the discovery progress going so the foundations can be built to be stronger, so the building to get cancer therapy working properly can be built higher.
Well, that's a great segue into the Block Lectureship, because what you've described requires collaboration all over the world by the greatest scientists who build upon what you build upon your foundation. And so we're gonna bring Li-Chin in a couple minutes, but first, explain the importance of collaboration and how it's helped you.
Science is a strange endeavor because it's actually both individual and also very communal. So we discover things and we like to think we personally have made a big contribution, but we only can make those big contributions if our own individual efforts are embedded and built upon societal efforts. And it's a sort of strange sort of combination of being individual but also communal. Communal in two ways. One is strict collaborations, as you said. Actually, I don't collaborate very much. I tend to work as a single lab.
You mean? Yeah.
But what I do is interact very strongly with lots and lots of different scientists who influence me in what I'm thinking, but I tend not experimentally to do joint experiments. I can do that, and I prefer to work that way. Most clinical investigations have to be big collaborations because of the complexities of what they have to do. But interaction is a word I like to add to collaboration, because if I didn't interact with people, if I didn't encourage challenge from other people, and if I didn't sort of keep an eye on what they were doing, then science would go much slower.
Wow. Okay. That was fascinating. We could go on for hours, but your time is valuable and you have 6 other things. So we're going to take a quick break, and when we come back, we'll meet Li-Chin and we'll hear more about the Block Lectureship.
The best outcomes for treating cancer come from early detection and early diagnosis.
The James Cancer Diagnostic Center at Ohio State
is designed to provide rapid evaluation and a clear diagnosis.
We are transforming care by giving patients direct access
to cancer experts who study and treat cancer every day.
If you think you have cancer, the choice is clear.
Choose the James Cancer Diagnostic Center.
Welcome back, and let's welcome Li-Chin. Thanks for joining us. And congratulations on being selected as the recipient of the Block Lectureship Junior Faculty Award. That's a great honor for young scientists. So fill us in a little bit on your research.
I study human genome organization and function in live cells. And then we use the technology that I developed years ago that we can look at how the genome is being organized in normal and versus cancer cells. And then we try to manipulate changing the important factors and then see whether we can try to reverse the mistake that has occurred in cancer cells, or sometimes when the cancer goes so badly that you cannot reverse it, then we use it as an identification tool to identify cancer cells.
So, Paul, you got to select the person who would be the recipient of this Junior Faculty Award. So, in Li-Chin's research, what is it you saw that made you say, hey, this person will be interesting to work with?
she's clearly a discovery scientist trying to discover new things, which is what I do. So I'm in the same territory, and so I thought my experiences might be useful. But more selfishly, I thought I might learn from her because I'm interested in what she's doing. And these sorts of relationships are reciprocal. I mean, they work best when you're learning something from each other.
Now, when you say you would learn from her, what is there anything you had in mind that specifically you wanted to learn, or just in general?
In general, she's using single molecule sort of techniques. These are important. I don't know too much about them. I know something about them. I know quite a lot about CRISPR-Cas and what that does, and indeed used earlier methods in yeast, 30 years before. But I have things to learn, so it makes it a nicer and more productive relationship.
What do you think you are going to learn from Paul?
Oh, a lot of things. So first, I'm a junior faculty. I just started my lab, about 5-6 years ago. I can learn a lot of how managing the lab, and also I think Sir Paul is very good to explain science to a broad audience, which I'm still learning that process. Not only the scientific community, you want them to understand your science, but also more broader, maybe the high school student, or as many people as possible. And I think Sir Paul is very, very good at it. And that affects how you write your paper, your grant, and also that makes people feel science is important. So those are more like a general things that I can learn. But on top of that, I think if I can discuss science with Sir Paul, that will be very impactful for my science, because Sir Paul has very unique thinking of scientific questions, so I can consult him about new technologies, and then whether my questions is a good question or not, or whether there's an easier technology that I can tackle specific question. I think there's a lot we can interact, and then I can learn from Sir Paul. Yeah.
I'm curious, how much did you know about him before, 3, 4, 6 months ago?
Well, I think his work has been written in textbooks. So there's no problem for me to learn that. I think I learned the earliest when I was a middle school student.
In middle school, you were reading about him. Wow.
Yeah, but maybe at that time it was translated into a simpler version for younger people. So
where did you go to middle school?
In Taiwan.
So your work is taught internationally in schools. Wow. Okay.
But I know much more because as the science goes, you have more details added into specific mechanism. And so when I was an undergraduate student, I learned the book is much thicker and then with more details. And then I believe there's at least one chapter is contributed by Sir Paul. There's a lot I can learn from Sir Paul to include it into my own research, that how genome has been organized in different cell cycle, and then how different genes can interfere with this organization and the function.
Wow, okay. So Sir Paul, the mentor-mentee relationship is a key to science. And before I ask you how your philosophy as a mentor, did you have a mentor who really helped you early in your career?
I didn't have somebody who was exactly like a mentor-mentee, as we're talking about here. But I had two supervisors who were enormously generous. When I was a first postdoc, when I was actually 23, 24, quite young, I went to two labs to learn two things. I worked with yeast. I'd never worked with yeast before. I went to a lab in Switzerland to learn how to do genetics in yeast. I then went to do my work in Edinburgh, in Scotland, to work on the cell division process. And both of those gentlemen, as Leopold and Murdoch Mitchison, devoted huge amounts of time just talking to me. They didn't see themselves, we didn't use the words like mentor then. And what I wanted to emphasize is they spent hours and hours with me, yet they didn't go on any of the papers I produced Why did they not do that? They said, well, we didn't contribute anything experimentally to those particular papers. It was an old school situation. This is in the 1970s. We don't do that anymore, but the generosity was huge, and I profited greatly, not only from what I learned, but also that generosity.
Wow, so that sounds like a little bit of a blueprint of how you will be a mentor, but anything you've learned since that you also apply to being a good mentor.
Of course. I mean, I think sometimes people use, mentors want to boss the mentee about, say you should do this, do that. And that is completely wrong, It's an interactive relationship to try and help work out difficult problems that requires two people treating each other equally. One may have more experience, the other will probably have more knowledge about the actual thing that they're doing. And it's that interaction, if properly handled, which is the most effective.
Someone has told me, a couple people have told me that young new scientists bring brilliant new ideas in that more established scientists may not have thought of, and that having someone to share them with to help you take that idea and move it forward is so important. And it sounds like that could happen for you.
So I think at the early stage of scientists, usually they're very ambitious. They will propose a very big ideas, and then in reality, that will probably not be completed in 50 years. And at that time, you really want someone tell you, this is too ambitious. You only need to do a portion of maybe 25% of it, and then you can complete it in 5-10 years. And you should go progressively to complete your big ambitious goal, not just initially set up a very high goal and then it can be easily failure.
Can I confirm one thing? You said 50 years. I've actually, the problem I set myself, it was almost 50 years ago, and I'm still working on it.
But that's to me is amazing, because it's not the exact same problem. It's changed and mutated 100 times, and it's always a little different. It's a giant problem that you're still cracking away at. So it's not one thing, it's hundreds.
It was quite a big problem, and there's many facets. But actually I'm driven by much the same objectives as I was when I was 24.
So what happens next? I know you'll be giving the lectureships, two lectures, and spending some time here. How do you envision moving forward with you two to share information and talk? I'm hoping we're going to meet in the next day, either tomorrow or the next day, to actually work out the best way, because clearly I'm about 4,000 miles away on the other side of a rather large ocean. So it's got to be a more remote relationship. But really what we need to do is to find out what is going to be most important for you, what is most useful to you, and then we can work it out from there. And we will find something, but I don't know what the way is. It's always unique, mentor-mentee relationships from one to another. And how do you see it going?
So I think nowadays communication is made easier than the last century, right? So we can do different ways like emails, phone call, as well as I can potentially visit Sir Paul in his institute. I'm considering all different possibilities, and then hopefully I can make it very efficient for both of us and very inspiring.
So Li-Chin mentioned earlier she's never been to the UK or Europe. So
That's
I
right.
think
Yes,
we need to change that and bring her over to your lab. You're in London?
I'm in London. We do speak the same language.
Yes.
Although we don't always understand each other on both sides of the Atlantic exactly. But yes, and it's a nice place to visit.
Okay, well, congratulations to both of you. It's been a pleasure to meet you, Sir Paul, and congratulations on the Block Lectureship, which is a wonderful, generous thing that the Block family has created 26 years ago. And Li-Chin, congratulations. I think this means you're headed in the right direction and have a great mentor to do great things.
Yeah, I believe so, and I'm very excited and looking forward to the interaction with Sir Paul I truly believe that he's going to give me good advices on my way, move to the mature scientist, and that will affect me a lot.
Me too. I'm looking forward to it as well.
All right, well, maybe in 16 or 18 months, we'll do this again in London. This podcast is brought to you by the Ohio State University Comprehensive Cancer Center, Arthur G. James Cancer Hospital, and Richard J. Solove Research Institute. For more information, check out our website, cancer.osu.edu.